the reproductive system, the first topic that will be dealt now is the sex differentiation and development. So basically reproduction is a process in which there is a perpetuation of the species. And when we say about human race, there is always an entity that we speak, that is sexual dimorphism. So silence please. So when we say sexual dimorphism, it just means that in the same species there are two sexually complete but different individuals. So both the individuals or two kind of individuals which are sexually different but they are complete in themselves. Then finally, why all this happens? The central theme of the nature is to preserve the species and perpetuate and cause the evolution. So what are the topics that will be covered? So here in this thing, sex determination, differentiation, the various abnormalities, and what are the methods of determining the sex of an individual. So the first thing, sex determination. So all of you familiar with the symbol?
Yes, sir.
Yes. So what is this?
Male.
The first one, male, and the other one stands for the female. So what does sex refer to? It refers to physical or physiological differences between male, female, and intersex bodies, including both the primary sex characteristics and also the secondary sex characteristics. So when we say about sex of an individual, it refers to both the primary and the secondary sex characteristics. So how are the sex differentiation happens? It happens at around 3 levels. So first thing, what the person appears, that is the phenotypic appearance. So what is the phenotypic appearance? Next, it is at the level of the gonads. So how the gonadal system has differentiated in an individual, and then at the level of the genes, that is at the level of genetic sex. So the individual can be determined for sex at the genetic level, the gonadal level, and finally it is at the phenotypic level. So determination is related basically to the genetic makeup. So what determines the sex of an individual is the genes. So what is the genetic makeup? That is going to decide what will be the sex of a developing fetus or the embryo. And differentiation, it is the subsequent steps that is next to the genetic sex. So once the genes determine the sex of the fetus, next all the embryonic structures get developed or differentiated according to the genetic sex. So that is the difference between the two things. So what all at the different level it happens? So one is the genetic sex, next happens at the gonadal sex, and then the phenotypic sex, where we speak about both the internal and the external genitalia. So genetic sex, you all know this, XX or XY. So when it is XX, it is the ovum or it is the female that is going to develop, and if it is XY, it is the male genotype. So we all have this. How many pairs of chromosomes? 22 pairs plus the genetic ones, that is XX or XY. So that is how. So if it is males, it is 44XY, and females, it is the 44XX. So this genetic sex will determine whether the embryonic gonad is going to develop into testis or the ovary. Now, what is this genetic sex? Why it has to be an XY or XX? What is the determining factor over there? Now, over this Y chromosome, there is a specific gene which is called as the SRY gene. What is being taught? Please note it down. Please note it down. The flow in which it is being taught, please note it down. The SRY gene, the Y chromosome has a gene which is called as the SRY. The name is sex determining region of the Y chromosome. Now this is present only on the Y chromosome, and this codes for a protein that is testis determining factor. So it is this presence of SRY gene which codes for testis determining factor determines the development of gonads into the testis. Okay, so this is the gene which is present on the Y chromosome. So usually we say if there is no Y chromosome, the fetus will become a female fetus. For example, if there is only one X chromosome also, instead of XX, even if there is one X chromosome, automatically the fetus becomes a female fetus. So only if there is a Y, or a saying, if there is a Y chromosome, it will automatically become a male fetus. Suppose you have, I think even in 12th you would have read some of the genetic abnormalities. Somewhere you got exposed, Klinefelters, Turners. So even if there is an abnormality where there is two X chromosomes with a Y, if there is a Y, it will become automatically a male. Or if it is a Turnus, that is only one X chromosome without a Y, it will automatically become a female. So it is the presence and absence of Y. Otherwise, a female fetus will automatically become a female fetus. So from the genetic sex, it has to determine now that it is going to become either testis or ovary. So next, the gonadal development. How will this gonad develop, and then from the gonad, the phenotypic development occurs. So both the testis and the ovary, they develop from the gonadal primordium. So the primordial structure or the embryonic structure from which the gonads are going to develop are the common ones. So whether it will develop into testis or ovary, it will be determined at 5th week of gestation. So till 5th week, it is undifferentiated gonad. It is a common embryonic structure. So from 6th week till then, till the complete gestation, the entire maturation and the differentiation occurs. So in gonadal sex, you can write, so till 5th week it is indifferentiated, and from 6th week only the maturation will start till the complete period of gestation. So this is like the 36 weeks of gestation where the fetus is inside the mother's womb. That is the period we are talking about. So 6th week, the maturation or differentiation starts. So what does this indifferent gonad actually have? So when we are saying indifferent gonad, it should have the cells for development of testis also. It should also have the cells which are meant for the development of ovary. So that is what is indifferent gonad. So this indifferent gonad has germ cells, it has Sertoli as a male part, or granulosa cells, which is the female counterpart. It has Leydig cells as a male counterpart, or the Tika cells. Okay, so these are the cells which are present in the indifferent gonad. So both the cell lines are present. So germ cells and also these granulosa, Sertoli or Leydig and Tika cells. Sorry. So the indifferent gonad, also called as the genital ridge. So indifferent gonad till how many weeks? 5th week. So 6th week, the maturation will start. So here the timeline you can note down. Sorry. So the seminiferous tubule with the Sertoli cells, that is the Sertoli cells in which the maturation of sperms occur. So that starts to develop at around 6 to 7 weeks. The timeline is so that the development in the males, that is the Sertoli cells or the Leydig cells in the males, it is a little earlier than the females. So the timeline will be a little ahead in the development of the male gonad. So 6 to 7 weeks, the Sertoli cells and the seminiferous tubule will start to form, and the Leydig cells around 8 to 9 weeks, and the Leydig cells are the cells which will start secreting testosterone. Okay. So what we want here is the gonads to start developing and the gonads to start secreting the testosterone. So this is what is needed for the development of the male pattern of genitalia. So first, 5th week is indifferent gonad, 6th to 7th week, some Sertoli cells will start to develop, Leydig cells around 8 to 9 weeks. Okay, so this is the timeline for the male gonad. The female gonadal sex, it will start only after the 9 weeks. So you can say the male pattern has developed, if not later on only the female pattern develops. So after 9 weeks only, the germ cells, they undergo and divisions to form the oogonia. So the characteristic is the female thing is the oocyte develops, okay? And when the ovum or the oogonia starts to form, that is starts after 9 weeks, the ovum starts to produce all the many ovums, or the oogonia starts to develop, they'll undergo many mitosis and meiosis. Mitosis, which increases the number, and then there will also be a series of meiotic divisions. But it so happens that the primary oocytes, they get arrested in after in the prophase after the first meiotic division. Now the primary oocyte, the oogonia develops, mitosis and meiosis happens. In the primary oocyte stage, in the first meiotic division, in the prophase stage, it will get arrested. It will remain as that till the girl attains her menarche. Okay? So this will be the stage of oogonia development, that is the primary oocyte arrested in the prophase of the first meiotic division, and it remains till the stage of puberty or the menarche. When the girl actually attains the menarche, this oocyte will complete this first meiotic and further series of development will happen. So this is the stage in the oogonia formation. But another thing that happens is, just look at it, around 4th month, that is 16th week, around you will have even more than 10 million number of oogonia. 28th week of gestation, 7 million, and at birth it is only 2 million germ cells. So as the maturation keeps on occurring, the number of oogonia will keep on reducing, and whatever oogonia are present, they are arrested in the prophase. Okay, so this is the stage of oogonia development in the female gonadal system. So you can say the female gonadal sex, the differentiation starts at 9 weeks. The germ cells, they undergo divisions to form oogonia. Now this oogonia will be arrested in the primary oocyte stage, in the first meiotic division, at the prophase stage, which gets completed only at menarche phase. And as the gestation progresses, the number of oogonia will keep reducing. At birth, only 2 million number of oogonia will be there, even though to start with it may be even more than 7 millions. Okay, so this you have to write. Please write it down. Write it down. Are you able to understand? So in males it's actually very simple. So you will just write about the Sertoli cells at 6 to 7 weeks. Leydig cells you write it 9 weeks. Female gonadal development starts at 9 weeks only, and the oogonia arrested in primary oocyte at first meiotic division, and then the number of oogonias will keep reducing. At birth only 2 million oogonias will be there. So this is the gonadal development.
The Leydig cells were already present in the primordial.
All the things. Actually, what happens? How it shifts over because of the Y chromosome, automatically this will start forming. The Y chromosome is not there, or
See, only if the Y chromosome is there, it will channelize for the development of male gonadal sex. In case the Y chromosome is not there, 6 to 7 weeks, 8 to 9 weeks will not happen. Automatically the switchover will, or the development will start by 9 weeks, and it will go into the female gonadal sex. So if there is a Y chromosome, it will start following the male gonadal sex pattern, and that timeline will start following.
I think more Leydig cells will start.
Yeah, that one only will start forming, otherwise it will get suppressed.
Thank you.
So next, once the gonads are formed, next it is the formation of the genitalia. That is both internal and external genitalia, in which the internal genitalia will form first and then the external genitalia. Now this internal and external genitalia, they need the presence of hormones. So the thing is, if there is testosterone, it will go for male pattern. Absence of testosterone, it is the female pattern. So that is why we say a fetus is conditioned to become a female fetus only the Y chromosome is channelizing it to a male fetus. So if there is, it's always about the absence of Y chromosome, absence of testosterone, that it is making it into a female fetus. So the internal genitalia, the primordial structures or the embryonic structures, it is the Wolffian duct, or the other name is mesonephric duct, which is for the male internal genitalia, and Mullerian duct or the paramesonephric duct for the female genitalia. All this, I mean, embryology, till where you have reached? Okay, so these are the primordial structures. Wolffian duct for male and the paramesonephric or the Mullerian duct for the female pattern. And external genitalia, again there is a common structure, that is the urogenital sinus, from which either the male or the female external genitalia can develop. So till 6 weeks or indifferent gonad, we say the fetus would have both Wolffian and the Mullerian duct. So when the gonad forms and the testosterone starts forming, automatically the female duct will regress and only the male duct will be present, and that will take the further development of the genitalia. So till 6 weeks, both Wolffian and Mullerian duct will be present in the fetus till the indifferent stage. So the Wolffian duct gives rise to epididymis, vas deferens, seminal vesicles and the ejaculatory ducts, and the Mullerian duct gives rise to fallopian tubes and the uterus. So why or what are the hormones that are needed? So we say that the Leydig cells around 9 weeks have started to secrete the testosterone. So now testosterone is present. Along with testosterone, there is another hormone, that is Mullerian inhibiting substance or the anti-Mullerian hormone. So a male fetus will have both testosterone and anti-Mullerian hormone. So these two will start or promote the growth of Wolffian duct, and MIS will suppress the Mullerian duct. So automatically the Wolffian duct maturation is promoted. Okay. So genetic sex, gonadal sex in a male fetus, Y is there. Automatically the gonads have to develop into a male gonad. So 9 weeks, Leydig cells is starting to secrete testosterone. So till that testosterone is there, it is both the ducts are present in the fetus. Now once testosterone and MIS are secreted from the male gonad, the Mullerian duct will regress because of MIS, and testosterone will promote the development of Wolffian duct. So from 9 weeks onwards, this maturation will start to happen. So what about in females? There is no testosterone. There is absence of Mullerian inhibiting substance. So starting around 11 weeks, there is a development of the Mullerian duct. So since there is no support of testosterone, what will happen? The Wolffian duct will regress. Right? So testosterone is not there, so automatically Wolffian duct will regress. There is no MIS, so Mullerian duct is not inhibited and the development will start happening. So here the timeline is around 11 weeks. So because gonadal development itself starts after 9 weeks. So you should think that till 9 weeks testosterone is present, means it will take the form of male fetus. So till 9 weeks nothing is happening, no testosterone. Okay, the female fetus now will start developing. So bipotential gonad, so Wolffian duct and the female gonad, so the two hormones, testosterone and MIS. Next we come to the external genitalia. So again the external genitalia needs testosterone, but one step ahead of that it needs the presence of dihydrotestosterone, that is the active form of testosterone. So this dihydrotestosterone, it actually gets, for the external genitalia, the target cells of the external genitalia will convert it into the active form under the influence of the enzyme 5-alpha reductase. So internal genitalia needs testosterone, whereas external genitalia also needs a active form of testosterone, which the conversion which actually happens in the external genitalia only, because it has the enzyme 5-alpha reductase. So testosterone to dihydrotestosterone is done by 5-alpha reductase present in external genitalia. So this form will start forming the masculinization will start around 14th week of the gestation. So till 9 or 10 weeks internal genitalia is formed, then at 14th week external genitalia will start forming in a male fetus. So external genitalia also needs dihydro with testosterone. So both the active form is needed for that. In a female phenotype there is no testosterone, so it will start forming into the external genitalia, but here it starts a little earlier because anyway testosterone is not there. So when the internal genitalia starts forming, external will also start forming along with that. But here in a male fetus it's a little late. Till internal genitalia the male fetus was a bit earlier, but in female fetus both internal and external starts to form together around the 10th week. So the embryonic structure for the development of external genitalia. So you can remember genital tubercle. Now for the male and the female, please note this down. Whatever has been taught till now, please note this down so that if a question comes, at least this much if you write it is fine. Right? So the genital, we have told that the embryonic structure remains the same, both for female and the male development. So the genital tubercle becomes penis, genital fold becomes the shaft of the penis, the genital swellings, they become the scrotum. Similarly in the females, the genital tubercle, they become the clitoris, the genital fold, the labia minora, and the genital swelling become the labia majora. So finally it is again testosterone or no testosterone. You can note this down. Fine. So, what are the steps of sex determination and differentiation? So, to start with, if it is the X chromosome and the ovum, and if it is the Y chromosome there. So, it's a sperm with Y chromosome, it becomes a male fetus or genetically a male. The embryo is a genetic male. So, the first step of genetic sex, and if it is female with a sperm with X chromosome and egg with X. So if it is XX, it is the genetic sex. The first step is genetic sex. Then the gonadal sex, where it is determined by the SRY gene present in the Y chromosome. No SRY gene, so automatically it goes into the ovaries development. Next step, once the gonadal of testis is formed, hormones are present. So the hormones will determine the phenotypic sex of both internal and external genitalia. So if the question comes on sex differentiation, you will have to write about what is sex, the presence of can say the different characteristic, physiological characteristic of an individual, which determines both internal and external genitalia, both the genetic reproductive organs and also the secondary sexual characteristics. Next you will write sex is determined at what all levels? Genetic sex, gonadal sex and phenotypic sex with internal and external genitalia. Yourself? Why not tell the timeline? Yeah. Yes, yeah. So each of this, now you tell me the timeline, how it gets differentiation, whatever is the step that was taught till now. So once, okay, the genetic sex is there, XY means male, XX is female. So what happens there? Tell me. So there is a XY chromosome, what happens? The next boy. If there is an XY chromosome, okay, the female or the fetus has got a XY chromosome and another fetus has got a XX chromosome now. What will be the next step of differentiation? So what will happen? Yes, what is that? Where? It is present where? Present in the Y chromosome, SRY gene present in the Y chromosome. So what does that do? So please what does it code for? SRY gene codes for which type? What is the protein? Testis determining factor. Okay, so what will that do? So presence of SRY gene or the testis determining factor, what does that do? What does that do? Because of SRY gene, what will happen? Next step, what it will happen? Masculinization of what? Of the fetus. So what will happen? Next step is what? After genetic sex, next step is what? Gonadal sex. So what is the timeline of the gonadal sex?
6 to 7.
Gonadal sex, what is the timeline that was told to you now? So before 6th week, what it is? Before 6th week, what is the state of the gonad that is in the fetus? Gonadal primordium. What is the type of gonad? Before 6 weeks, till 5 weeks, what is that type of gonad? What do we say?
Indifferential.
What was the term that was used? Indifferent gonad or bipotential gonad. When we say bipotential, till 5th week it is bipotential. That means it can either form a female gonad or it has the potential to form a male gonad. So till 5th week it is a bipotential gonad. So now Y chromosome is there, SRY gene is there. So now next what will happen? So 6th week what happens? What is the timeline? 8 to 9 weeks is for Leydig cells, testosterone and sertoli. What is the timeline for sertoli cell development? 6th week. So some 6th week there is a formation of sertoli cells and Leydig cells forms by 9 weeks secreting the testosterone. So next what about the female? Now in females the development of that oogonia, germ cells, everything, what is the timeline there? What? 4th month, okay. No, development starts at, not at 4th month, even before that. What is the week there? By 9 weeks the oogonia starts its maturation. And what actually happens? What is the property of that oogonia there? Does the number remain static throughout the period? No, it is actually it keeps on regressing. The number of oogonia, the pattern is to regress. So at birth it is only how many? 2 million. And what is the stage of development there? All this oogonia that is developed at birth, what are the stage? It is in the prophase, arrested in the prophase of the first meiotic division. And when does it get completed? Even this, it gets completed at puberty. So once the gonadal sex is formed, then what happens? You are still there? Yes. Yeah. Yeah. So once the gonadal sex formed, then what happens? 9 weeks, everything is happening. Okay. Now male gonad is formed. So what will happen? After gonadal sex, what is next to form? Genetic sex done, gonadal sex done. Next is what? What is it? What was that told to you? What is that? Pardon? Mullerian. Okay, but what is all that? What is that type of sex that was told to you? Genetic, gonadal and phenotypic sex. And now that phenotypic sex is both at the level of internal genitalia and external genitalia. So what is that internal genitalia going to develop from? What are the primordial structures? What are the two structures for male internal genitalia and female internal genitalia? Wolffian duct for males and Mullerian duct for females. So why does Wolffian duct develop now? What is the determining factor for Wolffian duct to form in a fetus now? Testosterone and Internal genitalia, we are not speaking about external genitalia. For internal genitalia, for Wolffian duct to form the male internal genitalia, what are the two things needed? Testosterone and Mullerian inhibiting substance. So testosterone for Wolffian duct, MIS or the Mullerian inhibiting substance to, what does that do? Inhibits or causes the regression of the Mullerian duct. It is a female counterpart. So that you don't need for a male fetus. So this happens around 10 to 11 weeks. And then what about in the female after all? That if at all a female internal genitalia has to develop, what is the determining factor there? How is it justified? Why does it develop into a female there? Absence of testosterone or absence of the Mullerian inhibiting substance. Then what about the external genitalia? What is needed for external genitalia to develop? So internal genitalia has formed. So next, the external genitalia. So what is needed for external genitalia? Testosterone and dihydrotestosterone. Because what is the enzyme? Pi-alpha reductase. So whatever the genetic abnormalities or the sexual abnormalities that we get to see, there is a chromosomal disorder also. That is basically if there is a discordance between these three levels of development. So in case the genetic development is not okay, then everything becomes abnormal. Or genetic sex is something else, but gonads do not develop. Again, there is a discordance between genetic and the gonadal sex. So what are all the abnormalities? So it could be either chromosomal, where there is a genetic problem, whether the XX or the XY chromosome, or it could be a developmental abnormality where the gonads may not develop. So some of the chromosomal abnormalities, Turner's, Klinefelter's, superfemales, and testicular feminization and hermaphroditism are developmental abnormalities in which you see. So what is this Turner's? X0. But since it is X0, what will be the phenotypic appearance? It will be a female because there is a X chromosome. Y is not there, so automatically it takes into the female development. But then there is abnormality like it will be a short stature, webbing of neck, cubitus valgus, broad chest, pigmented navy. Navy means a lot of moles will be there. Pigmented moles will be there. And coarctation of aorta, cardiovascular abnormalities, okay, cardiac abnormalities developmentally can be there. And then ovaries are not developed. It will be just like a streak over there. The gonads may not develop properly, even though it is X0, but since it is just X over there, even though it goes into a female gonadal development, the gonads development is not complete. It is not fully a mature gonad. But X0, so it goes into a female. Klinefelter's. What is the chromosomal abnormality? XXY. So what will be the gonad? Y is there. So what will be the gonad? Testis. So here, phenotypically and genetically, the male it will be a male because there is a dose of Y chromosome. It has to be a male. But again, abnormalities. So because there is an extra dose of X chromosome over there, so some of the features will be like frontal barrenness may not be there, poor beard growth, narrow shoulders, wide hips, female type pubic hair pattern, testis may be of smaller size. There may be sometimes gynecomastia or breast development may be seen because there is an extra ounce of X chromosome over there. Okay, so in both Turner's and Klinefelter's, one is a female, one is a male. One, the gonadal development is not complete maturation as of in a female in Turner's. In Klinefelter's, though a male, but an extra X chromosome creates certain female type characteristics. That's how we can remember. Super females, there is XX and X. So there is again the extra ounce of X, but since it is XXX, it will be a female pattern over there. Another one here, testicular feminization syndrome. Here actually what happens, it is XY. If it is XY, then male. The gonad has to be a testis only. But what happens? The gonads may be undescended, right? It doesn't get descended into the scrotum. So somewhere in the inguinal canal or in the abdominal cavity, the gonads remain there. So what has happened to the gonads now? They are not fully functional. Though the gonad is testis, though the genetic sex is properly XY, there is a developmental abnormality. So once the gonad are not fully formed, automatically testosterone may not be secreted well. So since the testosterones are not secreted, now what will be happening over there? The individual is feminine. Okay? The external genitalia is feminine. The breast development may be there. Pubic and axillary hair is scanty. The vagina ends in a brown pouch, but there is a absent uterus. So the individual will be phenotypically a female because testosterone is not there. So internal and external genitalia doesn't take the male development. So genetic sex is XY, that will determine the gonadal sex. So testis starts to form. But what happens? The testis is not fully functional. So testosterone is not secreted. So testosterone is not secreted means what will happen? Internal and external genitalia will not be formed. So that will be the developmental abnormality here. So therefore the individual phenotypically is like a female. But when the genitalia is examined for, it will be an abnormal genitalia there. There will not be a uterus in them. So what is this? Pseudo-hermaphroditism. So here the gonad is of one sex, but the genitalia is of another sex. So the testicular feminization syndrome is actually a form of pseudo-hermaphroditism. What is hermaphroditism? Bisexual. So both the gonads have to be there. But here we say it is a pseudo-hermaphrodite because the gonad is something else, the phenotypic or the genetic sex is something else. So in male pseudo-hermaphrodite, so the term when it is used, male pseudo-hermaphrodite, the male terminology stands for the genetic makeup. So when we say male pseudo-hermaphrodite, it means genetically it's a male, but the genitalia or phenotypically they are females. If we use the term female pseudo-hermaphrodite, it is genetic female, but genitalia or phenotypically it is of a male type. Okay, so that is a discordance. So genetic and phenotypic do not match here. That is one. So one example is testicular feminization syndrome, where genetic is XY, but internal or external genitalia is of a female type. Okay. Enzyme deficiency, actually this is with some of the adrenal cortex is done. Yes. So what is the enzyme deficiency that you can remember? What are the hormones from adrenal cortex now? What are the hormones from adrenal cortex? Glucocorticoid, mineralocorticoid and sex steroid. So what is that synthetic pathways? You remember that together? So if at all you try to remember what is happening, there is a pathway which puts it into the cortisol pathway, mineralocorticoid pathway, or it can get channelized directly into the sex steroid pathway. So what are the enzymes there? 17 hydroxylase or what is that? 21 hydroxylase deficiency. What will happen? See, what is that pathway which puts it into the cortisol pathway? It is one which finally channelizes into cortisol. 21 cholesterol is there. Progesterone will be there. Finally cholesterol. So one enzyme which puts it into the cortisol pathway, 21 hydroxylase. Now if cholesterol, this pathway is affected, it is not going into the cholesterol pathway, 21 hydroxylase is not there, the precursor is getting accumulated. So where will the precursor get channelized now? The other next pathway. And all these precursors, finally, if you look at it can get channelized to the sex steroids, which is testosterone over there. Androsterone, androstenedione, which are androgens basically. So there is another source of androgens apart from testis is adrenal cortex. So if the cortisol and all they don't form, it will finally go into the androgen pathway. So such enzyme deficiencies can lead to pseudo-hermaphroditism. Okay. So even though genetically they may be females, but if at all the female has a 21 hydroxyl deficiency, more of androgens will start forming. So this excess androgen will cause, what is the terminology that was used? Virilization. So more testosterone will cause virilization. So that is how one pseudo-hermaphroditism. So how is sex determined? One is karyotyping. So karyotyping is what? The chromosomal assay. So the chromosomes are arranged and you get to see the different varieties of the chromosomes. Another one is the bar body. What is bar body? Introduced? This term has been introduced? No. Any cell, bar body basically is any cell which has more than one X chromosome. If at all such a thing happens, one of the X chromosomes will get inactivated. That inactivation process is called lionization. Therefore, the hypothesis stands as Lion hypothesis. So this is seen in all the somatic cells. So this can happen, bar bodies can be present only in females because all the somatic cells, there can be one X chromosomes extra. So bar body is a determination for female sex. Right? So from today's class, the question can be the steps of sex differentiation or the determination. So you have to write about the genetic gonadal and the phenotypic sex, write with the different timelines, write about the embryonic structure, how it gets determined. That is important.